Hello, my name is Edlyn Morrow, and I am one of the members in Group C. Today, we will be discussing the following presentation on how do poxviruses replicate in humans. So, smallpox is a member of the viral family Poxvirus, and it originated from a species of the variola virus. The origin of smallpox is unknown. However, the earliest cases date back to 12,000 years ago during the first agricultural settlements in northeastern Africa. Sources claim that it spread from Africa to India by means of ancient Egyptian merchants. The earliest evidence of skin lesions, such as those that resemble smallpox, were found on the faces of Egyptian mummies from 11,000 BC to 1580 BC.
Now moving forward to transmission. Poxviruses infect their host by all possible routes. These following routes include the skin via mechanical means, the respiratory tract, and even an oral route. It's important to note that infection in humans usually occur to contact with three contaminated sources, thus being animals, people, or materials. Animal to human contact and transmission may occur via bite or scratch, meat preparation, direct contact with body fluid or lesion material, or indirect contact with lesion material such as through contaminated bedding. Human to human contact, however, and transmission is thought to occur primarily through large respiratory droplets. These respiratory droplets usually come from things such as coughing or sneezing. It is also critical to note that the most common form of transmission is through face-to-face, with the least common form being through airborne means.
Symptoms. So, smallpox is an acute and infectious disease, and there are several stages of symptoms. In the primary stage, this is usually the initial onset of symptoms, and this includes high fever, head and body aches, and some may experience vomiting. This usually lasts between 2 to 4 days. And when symptoms present during this first stage, the person may or may not be considered contagious. The second stage is the early rash stage, and this is considered to be the most contagious stage. It usually lasts about 4 days, and during this stage, a rash will begin to form on the person's tongue and or in any other area of the oral cavity. The third stage is the pustular rash and scab formation. This lasts for about 10 days, and it is also considered to be a contagious stage of smallpox. Those infected will present sores that become pustules, and after 5 days, the pustules will begin to form a crust and they'll start to scab. The fourth stage is where the scabs essentially begin to fall off the individual, and this can last for approximately 6 days. Finally, in the last stage, this is where the person is no longer contagious, as all of the scabs have fallen off and there are none remaining. Structure and genome organization of smallpox. So as we can see here, the viral structure of smallpox is enveloped with a double layered membrane, and this basically embeds a nuclear protein complex, which we can see like right here. The genome contains a linear double-stranded DNA, and the core of the virus, which is right here, is dumbbell shaped. So that usually has enzymes that can mediate gene expression. So nuclear proteins are also found in the core, and those are responsible for governing DNA transcription. So outside of the core here, this huge portion is considered the lateral body. So the function is not fully understood, but it's believed to be an area that basically acts as a storage unit for the virus. So it'll have important enzymes in there for viral replication, and it can release them when needed. So finally, I'm going to discuss the smallpox viral infectious cycle. So here's an overview. So as we can see, there are 3 stages involved, and this includes the early, intermediate, and late phase. So the early phase, which is right around here, is basically where the virus attaches and enters the cell via membrane fusion, and it starts to transcribe and translate proteins. This extends into the intermediate phase, where more proteins will be transcribed, right, and translated. And then the final stage is the late phase, which is a major portion here. And the late phase is basically where late messenger RNAs are synthesized, and those are responsible for producing structural proteins, right, and other enzymes that can be used to assemble virons or viral particles. So the viral particles, which start to form right around here, they have to undergo a process of maturation. And once they've fully matured, they can actually undergo cell lysis or exocytosis to leave the host cell.
So for attachment and entry, it's important to note that poxviruses actually produce two types of virions. They produce mature virions or MVs, which are responsible for host-to-host spread. And they produce extracellular virions or EVs, which are responsible for spread within the host, spread within tissues in the host. I'll be focusing mostly on mature virions for this. As for mature virions, they attach and enter through protein-mediated endocytosis. The first step in this is contact of the viral and host cellular membranes. Um, and that's mediated by four proteins: D8, which binds to chondroitin sulfate on the host cell membrane, A27 and H3, which bind to heparin sulfate, and A26, which binds to laminin. And then after this attachment, we see lipid mixing between the membranes. And finally, the formation of a pore. And this formation of a pore is mediated by an 11 protein entry fusion complex, or EFC. And we can see the proteins involved in this complex as well as their functions and properties in this figure right here. And then after the formation of this pore, once the viral core enters the host cell, the genome is released, and this triggers DNA replication. And when replication is triggered, the replication proteins are translated from early mRNAs by the host's translation machinery. And then after this occurs, we begin to see the expression of metabolic proteins such as ribonucleotide reductase or DPase. And then finally, we see the expression of conserved genes in the early phase as well.
For the intermediate phase, efficiency of the virus dismantling the host membrane is solely dependent on the presence of a plasma membrane outer coating. This is where the poxvirus sheds its outer coating and finally begins to replicate itself within the cytoplasm. Newly synthesized viral DNA molecules serve as sort of a template, if you will, for transcription. And this is why the initiation proteins from the early phase and cellular proteins are required for transcription of viral intermediate genes. During the late phase, this is where late mRNAs are translated to synthesize viral structural proteins, enzymes, and more. This is also where the assembly of progeny virions via viral factories are being had, while immature viral particles transition into maturity. Maturation and Golgi wrapping upon proteolysis and release from the bacterial factories is what drives the release through cell lysis or exocytosis.
Now to discuss the exit of a viral particle. Infectious poxvirus particles are produced in the cytoplasm, as can be seen in the image to the right. There are two mechanisms of release: exocytosis and cell lysis. In order to be released via exocytosis, these particles are further enveloped by intracellular membranes to form intracellular enveloped viruses that can then be brought to the cell surface on microtubules and exposed on the cell surface by exocytosis. Alternatively, the particles can be released through lysis. By definition, cell lysis is a method in which the outer boundary or the cell membrane is broken down or destroyed in order to release intracellular materials from a cell. This kills the cell but results in the release of viral particles. The eradication of smallpox. According to the CDC, the 33rd World Health Assembly declared that smallpox was eliminated on May 8, 1980. This is the first and only eradicated disease, and this was done using widespread vaccination. There are other poxviruses still in circulation today, such as monkeypox and Poxvirus, that we have yet to eradicate. Now to discuss the research and future directions of poxviruses, such as Poxvirus, in science. Research is being done to determine the suitability of Poxvirus to be used as an oncolytic virus. Oncolytic viruses are a new form of treatment for various cancers. Poxvirus may be suitable to be used as such because of its desirable safety and efficacy features, such as its mild symptoms, relatively slow replication cycle, and its inability to be spread human to human. It could be potentially used to treat metastatic cancer types such as breast cancer, ovarian cancer, pancreatic cancer, retinoblastoma, and melanoma. This diagram displays how oncolytic viruses can help regress tumors by infecting the cancerous cell, causing them to lyse. This concludes our presentation. Thank you.